Is about T helper 2 cells and extracellular pathogens. Helminths are too large to be phagocytosed, so mechanisms other than macrophage activation are needed for their destruction. Now here in this intestine there's a worm here, and this worm is too large, it's big size, so it cannot be phagocytosed by macrophages. However, there are in the wall of the intestine, there are cells like dendritic cells. These dendritic cells, they are able to recognize this parasitic antigens, and they can take them to the lymph nodes. So there are regional lymph nodes here as well. These regional lymph nodes, I'll draw a big one here. A big lymph node here to see that what happens during the process of activation. So these dendritic cells, they are going to enter these lymph nodes along with the antigen that they have captured with the help of pattern recognition receptor, and they brought them to the lymph nodes. So there are receptors on their surface. They recognize the pathogen with the help of PRR, pattern recognition receptor, or toll-like receptor, and then they chop them down into small pieces and then present it with the help of MHC class 2. Same time, they express, when the pathogen is recognized by pattern recognition receptors, the danger signals activate the expression of co-stimulatory signals, and these co-stimulatory signals are CD80 and CD86. So these are the co-stimulatory signals. Same time, if you think about the blood vessels have got T lymphocytes and B lymphocytes in them. The T lymphocytes, they are going to now come here, and these T lymphocytes will be presented the antigen by dendritic cell with the help of MHC class 2, and the receptor on the T cell that will recognize it is the TCR and CD4 receptor, CD4, and the co-stimulatory signal that will help it recognize is CD28. So there will be CD28 as well that will interact with CD80 and 86 on the dendritic cell. By these different signals, the first signal is MHC TCR, the second signal is the co-stimulatory signal, and the third signal is the expression of the cytokines. Certain cytokines are produced as a result of this helminth, and the most important one is IL-4. The interleukin 4 will be produced by this dendritic cell to activate these T cells. Now these T cells, which started off as a naive mature T cell, now these naive mature T cells have been primed. So among the millions of the T cells which are specific for this parasite, few of them that have now come, and these have been presented with their cognate antigen, and once this cognate antigen has been presented, these T cells will be activated. Now these T cells are activated. The T cells that come here, they are now activated, and how do we know that they are activated? They start expressing special receptor, and one of the important ones is CD25 which is actually the receptor for IL-2, and that's the beta chain, and they will start producing certain cytokines, and these cytokines include IL-2, and these cytokines will again act on themselves, and it will help in multiplication of these T cells. So these T cells will proliferate. These activated T cells will proliferate. This is called as clonal expansion. After clonal expansion, you can see there is a lot of IL-4 in the environment. There's a lot of IL-4 in the environment, and the presence of IL-4 will differentiate these TH cells into TH2 cells. T helper 2 cells. And these T helper 2 cells are now the effector, the differentiated effector cells. The signature cytokines that are produced by TH2 effector cells are IL-4, IL-5, and IL-13. I'll explain their functions in a while, but at the moment what we need to know is that these TH2 cells have become differentiated into effector TH2 cells and now they are going to produce signature cytokines like IL-4, IL-5, and IL-13. Now, looking at these B cells are also here. I'll just draw a big region of follicle, because normally the B cell rich area is the follicle of the lymph node. So this huge thing is the B cell area, and it is called the follicular region. So B cells are here, and B cells have got on their surface the BCR, B cell receptor, which is able to recognize the fragment of this parasite, and it is bound to its BCR, and it captures it, engulfs it or phagocytoses it, and chop them down into small pieces, and now they are going to present it with the help of MHC class 2, because B cells are also capable of presenting the antigen with the help of MHC class 2, and they will express the co-stimulatory signals through CD40. There is CD40 receptors which are expressed on these B cells. Now what these B cells will do, they will move to the extrafollicular region. They are going to come here. So the B cells have come here in the extrafollicular region. In the extrafollicular region, as I said, they are going to present the antigen with the help of MHC class 2, and CD40 receptor is expressed as a co-stimulatory signal, and these T cells, they are going to receive it with the help of TCR and CD4, and co-stimulatory signal will be CD40 ligand. And what will happen? These TH2 cells, as I said, they are capable of producing IL-4 and IL-5 and IL-13. This IL-4 and IL-13, they will send signals to this B cell to be differentiated into the B cells, and then further plasma cells, which are able to produce IgE antibodies. Normally the B cells in the beginning of any immune response, they produce IgM antibodies, but when the antigen is presented, the parasite antigen in this case is presented to these TH2 cells, these TH2 cells will send the signal in the form of IL-4 and IL-13, and the B cell will class switch. So there is class switching. There is class switching. It means that the heavy chain of the antibody they are producing is going to change from IgM to IgE. This is called as class switching, and these plasma cells are then start producing a lot of IgE antibodies. Now these both cells, they are actually short-lived plasma cells. They are short-lived plasma cells and memory cells. They can do this for a couple of weeks, not more than that. And there is a next stage to this process where this germinal center, it has got two regions. This is called as the dark region, and this is called as the light region. In the dark region, the B cells that have started proliferating here, they are going to enter, and they will be multiplying extensively. And while they are multiplying or they are proliferating, so normally their doubling time is around 6 hours. 6 hours is their doubling time. So in a week time, they can produce thousands of B cells. Thousands of B cells against this parasite. But they'll do one thing, a process that's called as somatic mutation. And this somatic mutation means, I'll just draw the receptor portion, this variable portion is going to change. Every time they divide, it's going to slightly change so that they get the perfect fit with this parasite antigen. And lots and lots of B cells will be produced. At the same time, if we see these Th2 cells, they have now moved to these germinal center as well, and when they move here, they are now termed as T follicular helper cells. These are called as T follicular helper cells. And there is another group of cell here which are specially present in the germinal centers, and these are called as FDCs. Follicular dendritic cells. Follicular dendritic cells also will capture the antigen with the help of the antibody receptor, FC receptor, or with the help of complement receptor. They will recognize the parasitic antigen, and their job is to present the antigen again to these proliferating B cells. So these extensively proliferating B cells, they will one by one enter the light zone. They will be presented with the antigen by the follicular dendritic T cells, and if the fit is perfect, then the follicular dendritic cell will give them survival signal. But if the fit is not good, these B cells will be destroyed, or they will die of apoptosis. They will die. But just a few which have the perfect fit, which are very strongly bound, they will be survived. They will survive, and these B cells are once again going to present the antigen with the help of MHC class 2. This time to these follicular helper T cells. And these cells are going to again give them the signal back in the form of IL-4 and IL-13. And as a result, what will happen? Now the B cells that are produced, they will be differentiated into, so here I am drawing the final B cell that survived, and it has got now the perfect fit for the parasite. And this whole process is called as somatic mutation and differentiation, and these B cells are now capable of producing the perfect fit IgE antibodies, which have already been class switched. Now these class switched antibodies and B cells, they are long-lived, memory cells, and plasma cells are also long-lived. So now these B cells and these Th2 cells, they are going to go back to the cell wall where this infection has occurred. So we assume that this is the class switched B cell and this is the Th2 cell along with its IL4, IL5 and IL13, and the B cell along with its antibody IgE and the plasma cell. They have arrived here. Now here is another cell that has come into play, and that cell is called as eosinophils. So these eosinophils are a granulocyte, part of the innate immune response, and these eosinophils have got special receptors, Fc receptors, and these antibodies which have been produced by the B cells, now these antibodies are going to bind to the parasite, and with the help of these Fc receptor, the eosinophils will recognize the presence of the parasite. So here is another eosinophil. This will recognize the presence of this parasite and they will start producing toxins, and eosinophils produce a lot of toxins. I'll mention some of them, like major basic protein or cationic proteins, or the neurotoxins. All these will help in the destruction of this parasite. Other than these eosinophils, there is another cell also present in the tissues, and that is called as the mast cells. Again, that is a granulocyte which is capable of producing a lot of mediators like histamine and prostaglandins. Histamine, prostaglandins, leukotrienes, and they will all enhance the inflammatory response. So the inflammation will be strongly induced by these presence of these mast cells, and eosinophils will directly kill the parasite, and when they are killing it with the help of antibodies, this thing is called as ADCC, antibody dependent cell mediated cytotoxicity. And as a result of the histamine, the eosinophils, there will be damage, and then these other cytokines, these IL4, they will activate these eosinophils, and IL13 will act on the epithelial cells of the intestine, and there will be a lot of mucus production, and they will increase the peristaltic movement of the intestine so that these parasites are expelled out. This is done by IL13, and the mast cell will release histamine and prostaglandin leukotrienes, which will help in increased inflammation, and these all things together will help in the damage to this extracellular pathogen or parasitic worm. When this all is going on, IL4 is going to act on another cell which is present here, and that is the resident macrophage. But this time, this resident macrophage is under the influence of IL4 is going to change into M2. Normally the M1 that classically we see is different from these alternate macrophages, which are called as M2, and this is under the influence of IL4 produced by Th2 cells. So when Th2 cells produce IL4, this is going to change these macrophages into M2, and their classic features are they produce IL10 and TGF-beta. And these IL10 and TGF-beta has got anti-inflammatory response. In fact, they help in tissue repair, and dampening the inflammatory response. So once this parasite has been destroyed, there is a lot of tissue damage, and to control this tissue damage, M2 cells are activated by IL-4. They will produce IL-10 and TGF-beta, which will help in dampening the tissue response, and it will help in tissue repair and fibrosis. So that's all about the T helper 2 cells and extracellular pathogens, and thanks for watching this video.